Oh, more. Yeah, now you guys are gonna be caught in 4K.
4K audio.
Yeah, 4K audio.
Starting now? Like already?
Like now. Okay. Okay. Can you see this? Why is this here?
It's a little Midwest.
It's fine.
I'm a boomer. I'm sorry.
I know.
Okay. Okay. So that chart that I gave you guys about Enterobacteriaceae, I will tell you, you should memorize this like through and through. Memorize it forwards, backwards, sideways, upside down, reverse. Just remember it. This is going to be your lifeline when it comes to Enterobacteriaceae. Okay. I would also remember the characteristics down here when it comes to boards and, well, this exam too. These characteristics right here are going to help you rule in something that is Enterobacteriaceae. So these are going to be very, very, very important. And mostly any boards question that you're going to get that's going to have one is going to have some combination of these. Okay. So lifeline, circle it, star it, rewrite it a million times. I think I've got blank versions of this that I have in the past given to you guys, just because it's so much information to remember all together, that you can just fill in right before the exam. Cool. All right. Also, I warned you guys about this. This is a really long lecture. We'll take a break somewhere in the middle of this, just so that you guys can like not be here. So, but feel free to get up at any point. Okay. So, Enterobacteriaceae. So I will tell you also the name has changed for this group of organisms. It's no longer Enterobacteriaceae, it's Enterobacteriales now. I'm unsure if boards will have that new nomenclature quite yet since it is relatively new, but you should know both terms kind of interchangeably, okay? Okay, if you remember nothing else from this lecture, down there on the bottom right of your chart are these 5 characteristics for Enterobacteriaceae. I would remember them. This is what includes them inside of this group of organisms. So the first one is that they're both gram-negative facultative rods. So like what we see right here, gram-negative rod, nice and pink. They all ferment glucose. So we have a number of ways that we can measure glucose fermentation. The big one are going to be these OF dextrose tubes. You guys remember these ones from your media lecture. Every single one of these bugs are going to follow this fermentative pathway. And this will kind of be a good way to separate out these gram-negative rods from others. For number 3, they're all oxidase negative, with the exception of Plesiomonas, because as you guys know, everything with micro, there is an exception. So oxidase negative. The other thing, number 4, they all reduce nitrate to nitrite, but never down to gas. So that'll be a good way to discriminate between Enterobacteriaceae and things like Pseudomonas aeruginosa, which go all the way down to gas. So that's a good way of doing that. They will also always grow on both blood and MacConkey agar. So some other gram-negatives might just grow on chocolates, that may just grow on blood agar, but with these ones, they grow on both. Okay. Sweet. So this is all the organisms that you'll be responsible for knowing for this lecture. It's substantially longer than any of the other lectures, but just kind of have it in one place. This is all outlined in your objectives as well. Okay, so before we get too far into this, I wanted to go and talk about a few media types because we'll be referring to them quite a bit in this lecture and in others. So first thing here, EMB agar, which, did you guys get to look at the EMB agar on your media lecture? Okay, awesome. So basically what I want you to know about this for EMB agar is I want you to know that it's selective and differential. I include some other things on here just in case you're a nerd like me. You can kind of like learn why it's selective and differential, but I just care that you know that it is selective and differential. And then I would also know like what it's good for. So it's really great for gram-negatives. The way I kind of think of EMB agar is it's kind of like a substitute for MacConkey agar. So if we were to have a national shortage of MacConkey agar, we would go to EMB as our replacement for that. Okay. And the great thing about this is lactose fermenters have like this black sort of greenish metallic sheen on here. And then the weak lactose fermenters have like a pink color and the non-fermenters are clear agar. So What's really cool about this, so with E. coli, which we'll talk about today, there's some defining characteristics when it comes to its growth on McConkey agar, like what it looks like, how it behaves, how it reacts with the agar. On EMB agar, it's the same. It has this really cool, bright metallic green sheen, like you see right here for the Christmas tree, and that's usually suggestive of an E. coli species. That's that lactose fermentation taking place there. Cool. HE agar. I love this agar because it does so many different things. I would know that it's selective and differential, and I would know that it's good for the detection of different pathogens in feces. So mainly Salmonella and Shigella are going to be the two main ones on here. We use this routinely in our stool cultures, and it's really got these combinations of reactions that you guys see here on this plate. So we've got this one right here that's no growth, so it kind of shows you what a no growth looks like, versus something that's clear. So clear right here suggests that it's not a lactose fermenter, and this would be suggestive of like a Shigella species. Okay. There's also this one that looks black on here. This is going to be indicative of H2S production. So this is going to be like what we would see something of suggestive of Salmonella species. Okay. There are other things, we'll talk about them today too, that are H2S producers that can also look like this, but this is usually like our first clue. Okay. Good. This is what we want. These are our lactose fermenters. These are this bright orange or salmon looking color here. This is what all of our E. coli is going to look like on there. Klebsiellas, all that wonderful stuff. And it is truly this bright. So cool. HE agar. McConkey agar. You guys will become very familiar with this one. We use it on most of our cultures in the lab for just routine setups. I would know it's selective and differential. And for this one, I would be a little more concerned that you know why it's selective and differential. It's selective through way of bile salts, so it inhibits gram-positives through that. And then it uses crystal violet as kind of the color change that's occurring there. And then it's got lactose in there to show different colors with it. But this is going to be great for isolating out different gram negatives. And this is what all the reactions look like here on the plate. So if we have a negative for lactose fermentation, it's going to appear clear on here. They have different morphology. Some of them like spread out. Some of them are really small and pinpoint. There's ones that are lactose positive right here, which are kind of this pink color, which kind of mimics the color of that agar. And then over here, this is also lactose positive, but if you look around the edges around here, you see like a little halo developing around those colonies. That's what we call a bile precipitant that's forming out of that. And this happens for E. coli because they're so efficient at metabolizing lactose on there that they're actually physically lowering the pH of that agar to the point where bile salts are precipitating out. And that's what we're seeing right here. And this is something that is really unique to E. coli. So like a bunch of ones that we'll see will follow this pattern right here. Cool. All right, there's another one, another McConkey agar called McConkey sorbitol. We call it SMAC agar for short. Really, the only difference is that it has sorbitol instead of lactose as the sugar that's there. And really, we use this as a screening agar for this E. coli 0157H7. So this is that Shiga toxin producing E. coli. We'll use this routinely on our stool cultures. And we'll talk a little bit more about E. coli 0157H7 here in a little bit, but basically it's bad, causes diarrhea, which, by the way, we're going to get super comfortable with poop in this lecture.
Cool.
Okay. And this is what it looks like here. So regular old vanilla E. coli, something that is considered normal for you, is going to appear like a sorbitol fermenter. It's going to be pink, not necessarily have a bile precipitant around it, but it's going to be pink. And then this E. coli 0157H7 is clear over here. Cool. XLD agar, which you guys had this as well, correct? Yes. Cool. All right. For XLD agar, know that it's selective and differential. If you're a super nerd, like take a look at all the ingredients in this thing because it's pretty impressive. But the way I would think of XLD agar is kind of like a substitute for HE agar. Okay, it does the same sort of thing. It's looking for stool pathogens, mostly looking for Salmonella and Shigella, just because of the way that they turn on the plate. So up here is kind of an example of that, where our Salmonella species are going to turn black, just like we would expect on HE agar. And then Shigella species are going to be clear or a pink color on there. And that's really all we would use XLD auger for. It can do other things, but that's all we care about. Okay, so any questions about media so far? Is that a good refresher for some of that stuff? Okay. So we're going to talk about some other important things when it comes to these bugs, things that make them pathogenic, things that influence their structure, and yeah, we'll just kind of talk about some other things. So cell wall structure, so knowing the difference between a gram-negative versus a gram-positive organism, you guys remember this, correct? Gram-positive, they have those really thick peptidoglycan layers on the outside of them. Gram-negatives, they are two lipid bilayers with a very thin piece of peptidoglycan squished in between the two of them. And this is really important because this is going to help describe where we're going to see the O, K, and H antigens inside of there. So it'll kind of give you a frame of reference of where they're located. Okay. The other important thing about Enterobacteriaceae is the use of a flagella. So you guys remember from your micro classes what a flagella is? It's the little wiggly tail on the end of the thing that causes it to squirm and wiggle through the environment. So most of Enterobacteriaceae can have a flagella, but there are a few that don't. And I will tell you, the easiest way to remember this is their initials right here, which is KS, or if you want to think of it, keep still. They don't have a flagella, they can't move. But it's for Klebsiella and Shigella species. Both of those are the non-modal organisms that we're going to be concerned with. Yersinia kind of has a unique thing to it as well. It's only mobile at room temperature, so if we were to do it inside of our regular incubator for a regular culture, like we would expect everything else to be modal at, this thing would not be modal. So can you not make so much noise?
Calm down.
Sorry for whoever's listening to this recording.
No, whatever.
And then, have you guys talked about Listeria yet? Okay, awesome. So then you guys know Listeria has that umbrella sort of shape motility. And this is what it looks like inside of here. Which Listeria is scary, scary, scary. There is a Listeria outbreak currently, I think, in the northeastern part of the United States, related to lettuce right now. So,
Oh, yeah.
Yeah.
My brother thinks that he and his wife got it.
Yeah.
To go.
Yeah.
Yeah. How long has that been going on?
It's been going on for a minute.
That's what I was thinking.
Yeah.
Okay, sorry.
Listeria outbreaks tend to last for a long time. If you guys remember, I don't know, I think it was maybe pretty close to 10 years ago now, there was the Bluebell ice cream Listeria outbreak. It was one of those that they pulled all the product off the shelves, they went back into production, found out Listeria was still in the ice cream, and had to pull it again. So it's one of those that's really, really persistent, and it's because of the temperature that it likes. It really loves really cold temperatures, and especially with ice cream, I mean, you're keeping it at a cold temperature. It's happy. It doesn't want to go away. So it's kind of a bitch to get rid of.
Right.
The other important thing about Enterobacteriaceae are toxins. A bunch of these bugs, every single one of them are going to have an endotoxin with them. So an endotoxin is something that's integrated into the cell wall. We call this the O antigen here for Enterobacteriaceae, and we'll kind of talk about that a little bit more here in the next slide. But every single one of them have that, and it's lipid-based. An interesting fact about the O antigen, it is one of the most potent things that we know of that will activate our own immune system. So when it comes to a patient presenting with a septicemia, a lot of times these patients will present in sort of like a shock sort of phase because if they have a gram-negative infection, that O antigen is causing them to go into that shock phase. Whereas if you're talking about a gram-positive organism, if you're talking about staph, a lot of them are going into hypovolemic shock, so their blood pressure will drop really, really quickly because they're forming clumps inside of that bloodstream. So it's kind of an interesting way of how our physiology plays with their physiology. Exotoxins, these are things that are protein-based. They're going to be from within the cell. They're going to be things that they are releasing into the environment. So when you think of these, think of, Salmonella is releasing an exotoxin, and Shigella releasing those toxins as well. And then enterotoxin. Enterotoxin could really be either an endotoxin or an exotoxin. That's kind of a misnomer right there. But it's just anything that acts on the intestinal tract is going to be considered an enterotoxin. Cool. Okay. So let's talk about our O antigen here. So we were kind of talking about this before. I would know the highlighted things on here. So the somatic antigen is the other name for the O antigen. A lot of times they'll throw that name at you just to, see if they can trip you up. But just know that they're the same. Hold on, I need to put my watch on Do Not Disturb because the group chat going crazy.
Is it micro load?
No,
No.
it's about a puppy,
Ooh, what kind?
German Shepherd. Okay, so O antigen gets its name from a German nomenclature. So Own is the German name for it. I think that's how you say it, but that's where the O antigen thing comes from. I would know that the antigen is also heat stable, and it's composed of lipopolysaccharide. So it's mostly lipid based. So in my little diagram down here, there is peptidoglycan right here, and these are our two lipid bilayers. That LPS, or the O antigen, is the thing that kind of extends off into the environment, the thing that's on the outside of the cell. Okay. Cool. K antigen, the other name for it is the capsular antigen, and again comes from a German name nomenclature for capsule, but you have to say it super aggressive to be German. And I would know that this is heat labile, so if we were to heat it up, it would be completely destroyed. And this is something that is only on encapsulated strains. So if they don't have a capsule, a lot of time nomenclatures will leave out the K antigen. Okay. And I would know that it's made out of made out of polysaccharides, so it's made out of sugars almost entirely. The other thing I would know about the K antigen is that it is part of the VI antigen for salmonella, okay? Which we'll talk about that with salmonella here in a little bit. Okay, and here is your German thing for the day. I don't know German. Do you guys know German? Anybody? No. I hear if you just yell words, it sounds like German. So, okay, H antigen. So this is going to be our flagellar antigen. It comes from the German name Hauch. It's heat labile, and only modal strains will have this. So which bugs would not have the H antigen? Exactly. Yeah. So we would always expect them to never have an H nomenclature when it comes to them. Okay, and this is composed mostly of protein. And these are our little flagella here on the end. A lot of these bugs have paratrichous flagella, meaning that's all the way around them. There's some that are on both ends of the cells, but these ones, if you were to take an electron microscope of this and get a high enough resolution, they would look like little hairy cells because they have so much flagella all the way around. So if you wanted a way to study this in simple terms, I love charts. I love making this as simple as possible. These are the things that I would know for your exam. The O antigen, the common name, is it heat stable or not, and then what it's made out of. Okay. And just to kind of show you how these things all kind of interplay here. So this, like I was mentioning before, comes with nomenclature. So this O25H5K2 here, this is how we would spell those out. So remember we were talking about E. coli O157H7. That's what we're designating here. And these are different serotypes of this bug. It kind of helps trace lineage of them, where are they coming from, and kind of do a contact tracing sort of deal when it comes to those bugs. But this is the order they'll do it in: O, H, and then K. Cool. And these numbers mean absolutely nothing to you or I. They mean something to an epidemiologist. So, cool. All right. These are some other biochemical things that we'll talk about throughout this. This lecture is very biochemical heavy. So Just kind of keep that in the back of your brain, but we'll kind of talk through these things as we bring them up. But just to kind of give you guys an idea. So this is our decarboxylation reaction here. And I want to kind of make you guys think about this, because the way I want to describe this is that these reactions are kind of interchangeable. If you have a decarboxylation reaction, no matter what your amino acid is there, the reaction is still going to appear the same. Okay. So for example, here, this amino acid here is arginine. If it's negative, it's going to remain yellow. If it's positive, it's going to turn purple. Makes sense? And you can go into the media characteristics of this, but this is a pH indicator. It's turning more basic here, causing it to turn a purple color instead of an acidic yellow color. Makes sense? So if we were to do like lysine instead of arginine, the reaction would be the same, just the base is different. Okay. This right here, this is an example of our urea test. So when I say something utilizes urease, we're going to have this bright pink color that shows up right here. And when I say urease, what do you think of? Yeah. So a good rule of thumb is if it's urease positive, it's probably a pretty good urinary tract pathogen. Okay. Yeah. We're just getting started. This is great. These are our TSI slants. You guys got to see these, correct? So knowing these, what's in the slant versus what's in the butt, what black means, that sort of stuff. And then do you guys remember the MVIC tests from your micro classes? It was like this.
We went over indole.
Did you? Okay. So, okay. Indole, just to make things complicated because we're microbiologists, there's two versions of indole. So this is tube indole, and then there's spot indole, which we'll talk about as well. But the color reactions are different, so we'll talk about that when we get there. But this is tube indole here. This is our methyl red. Methyl red is just referring to a different way of looking at sugar fermentation. And honestly, this is a garbage test. I would never require you to know that, nor would boards. They would just want us to talk about it. Folks Pascauer, another garbage test. And then the citrate test right here is whether or not they will utilize citrate. And there's only a few bugs that actually do that. But we'll talk about those when we get there. Okay, cool. So biochems, man. All right, we're gonna jump into the bugs. Any questions about the stuff we talked about so far? Nah. All right, let's talk about E. coli. Or Escherichia coli, if you want its government name. So on blood agar, the most common type of E. coli, and this is such a bad picture of it, but when you Google things, you get what you get, right? So for typically we would expect this to be a beta hemolytic colony. We would expect it to start eating through that agar. That's going to be our typical E. coli, okay? But they can range in any number of morphologies. They could be non-hemolytic. They can be super mucoid to the point where they look like boogers or slime dropping down the plate. Sometimes they are even really, really small and they don't want to develop super well. But what I would know just about gram-negative colonies altogether, if you have the typical case, is most of the time they're going to be large, spread out, flat, gray colonies. Okay, much bigger than gram-positives. So McConkey here, this is the other agar that we'll want to pay attention to. It is a lactose fermenter. See that pink color that's developing there, and you see this bile precipitant that's kind of forming around it in this grainy picture. But this is what we expect out of the typical E. coli, is that it's a lactose fermenter with a bile precipitant forming around it. Now, I will tell you, not all E. coli follows this. Some of them are non-lactose fermenters. They lack the lac operon altogether. They're a mutant, or they lack parts of a functional lac operon, which makes things even crazier. So, cool. E. coli. By the way, when you say E. coli, how many of you guys have like a negative connotation of E. coli? Like you just associate bad things with E. coli? Okay. Well, good. It's not everybody, because I feel like in the news and everywhere, E. coli gets a bad rep. But really, E. coli is a normal part of your GI flora. It is something that we want to be there. If it's not there, usually it's going to be causing you some sort of issue. Okay. It is actually the most isolated bacteria in the clinical lab. So when you guys are on the urine bench, this is going to be your #1 customer. We run into E. coli all the time. And oftentimes it's an opportunistic pathogen, meaning it's not something that's intending to cause you harm, but it found the opportunity and took advantage of it. So if you guys remember the difference between endogenous and exogenous infections, this is usually an endogenous infection. Okay. It's the most common cause of UTI, and we kind of talked about the colonies. But something you'll want to remember about this is the quick identification for this. So we rely a lot on MALDI-TOF, but if this, like I was saying, has a beta hemolytic colony on blood agar, it's a lactose fermenter with bioprecipitant, you could follow it up with this indole test here. We would do a spot indole test, and we would expect for it to be indole positive, which is what I have here. Okay. So this is a good time to talk about the difference between spot and tube indole. So spot indole, we are just taking a swab, picking up a colony, and then we're dropping a little dropper of indole reagent on there, and we're going to expect a blue to be positive like we see here. No color or like a red color is going to be negative. Okay, and that's different from our tube indole. Our tube indole, we use Kovacs reagent, which if it turns red, that's a positive result. And if it's no color change, it is negative. So there is a difference. All right, other flavors of E. coli. There's E. coli O157H7. There's enterotoxigenic E. coli, enteroinvasive, enteropathogenic, enterohemorrhagic, and then a condition known as hemolytic uremic syndrome. Okay, so we'll kind of talk about these. So E. coli O157H7. This is the bad E. coli. So when you hear about those, well, Chipotle, for example, this is the E. coli they're talking about here. Okay, it produces a Shigella-like toxin. It's a toxin that's very, very similar to one that is in Shigella. Which, how many of you guys played the Oregon Trail when you were a kid? Please don't tell me we're beyond that.
You're not.
Okay, okay. How many of you guys died of dysentery?
Many times.
Yes, Shigella is the bug that caused it. So, but basically, the toxin works by inhibiting protein synthesis and just locks open these chloride channels. Okay. But it's kind of interesting. This is something that lives inside of cattle, swine, and deer, so it's mostly associated with livestock. But they do not have the receptors to like get any sort of illness from it, which is why they have no signs or symptoms before we ingest that product and then eventually get sick. So cool. So E. coli 157, we've got a couple of ways that we can detect for it. We check for the toxin itself on a little lateral flow assay. It looks like a little pregnancy test where we're looking for just the Shiga toxin producing E. coli. We also use our SMAC auger, which is kind of what I'm mentioning here. This is the way we'll screen for it. We also have PCR tests that we can look for as well. Cool. Enterotoxigenic E. coli, I would associate the term traveler's diarrhea with this. And it's a major cause of diarrheal disease in lower income countries. So if you think about, you know, how when you go down to Mexico, they tell you to avoid drinking, you know, like tap water and all of that stuff whenever possible. This is usually one of the bugs that can cause it, other than a few viral illnesses, but enterotoxigenic E. coli is a customer there. So it's usually transmitted via food or contaminated water sources by human or animal feces, which is kind of gross to think about. But the infection can cause profuse watery diarrhea and abdominal cramping, and usually lasts 1 to 3 days, and sometimes can extend to that 4th day. But it's one of those that's kind of like hits you really hard and then just kind of disappears. So most people go underdiagnosed for it. Most patients will recover by supportive measures, so they'll just tell you to drink Pedialyte or something like that, and usually they won't need any sort of antibiotics or anything as an intervention. And there's a toxin that it produces that acts on the intestinal lining that causes that diarrhea. Okay. That's in contrast to our enteroinvasive E. coli, which will basically just like wiggle its way through your cells and cause kind of an irritation, which causes that diarrhea. But this is one of those that we can't actually culture for. This is one, well, I take that back. We can culture for it, but we won't do it on agar plates. We'll do what we call, have you ever heard of a tissue culture for like viruses? Is that term familiar with you? Basically, we take a lineage of cell lines. So we'll take something like your intestinal cells or something like that, put it inside of a liquid media, and then we will inoculate a specimen into it and look for some sort of cellular destruction. And we'll talk about this when we talk about virology. But that's the only way that we can culture for this Enterobacteriales E. coli. So most of the time, it's ID versus through PCR. Enteropathogenic E. coli. I would associate this with newborn nurseries. This is one of the causes of diarrheal outbreaks there. I will also tell you this is one of those that's kind of like norovirus in the virus world, where it kind of hits you for a day and then disappears. So a lot of times it's underdiagnosed, but usually we'll use PCR to ID this one here. Okay. So enterohemorrhagic E. coli is a term that we use to describe E. coli 0157H7 because it causes kind of bloody, profuse diarrhea. And it can cause kind of this thing called hemolytic uremic syndrome, which is basically the abnormal destruction of red blood cells. And when you destroy those red blood cells, your kidneys are trying to filter it out, and ultimately you clog them up, building up an excess urea inside of your system. And that can be deadly, which is why we also tell people to avoid antibiotic use with this whenever possible. So antibiotics will kind of promote that. So, cool. And this is what that little lateral flow test looks like when we're looking for that Shiga toxin producing E. coli. Super cool. And this is a chart comparing all of them. So if you're really interested on like where they act inside of the body, you know, whether it's the small intestine, large intestine, please by all means take a look at that. And then a little more on pathogenesis over here and the disease state that it causes. Questions on E. coli? No? Okay. Shigella or Shigella. So blood auger more or less looks the same as our E. coli, just kind of this large gray sort of colony. And typically these are going to be non-hemolytic on there. Conky auger, these are non-lactose fermenters, so they're going to be clear or sort of this yellow appearance on there. And then the big auger that we're going to want to pay attention to is this HE auger. So this is the way we're most of the time going to catch this. It's going to appear like this green or translucent colony on there. No lactose fermentation is occurring, which is why it's that green color. Cool. Shigella. So the infection is caused by an ingestion of contaminated food or water. It's extremely infectious, so it requires less than 100 bacterial cells in order to cause a full-scale infection. So if you think about that, that's not even the size of a colony for you to ingest for that infection to begin. So virulence factors include invasiveness of intestinal lining of the large intestine, causing cell death and sloughing of the lining. So occasionally people with this sort of bug, we've seen it before where we get stool samples with parts of their intestines inside of there because the inside is sloughing off. So that's not something that's completely uncommon. But it produces an enterotoxin. It's the OG Shiga toxin. This is where it all started. And it causes dysentery, which is the inflammatory disorder of the lower bowel, causing pain, fever, severe diarrhea, and often accompanied with passage of blood or mucus. So it is very, very uncomfortable. And by the way, I wanted to define what diarrhea is for you guys. It's 3 or more loose stools over the course of a day. So that is the medical diagnosis for what diarrhea is. And so people with Shigella are oftentimes having profuse, watery, bloody diarrhea about 10 to 15 times a day. It's pretty bad.
Where would you get Shigella?
Well, it's kind of interesting. Shigella kind of works in waves. So Shigella, around every 5 years or so, we experience an outbreak in this country. But we can get it from contaminated food, so we can get it from contaminated meat sources, we can get it from contaminated vegetable sources, things like that. To my knowledge, we have had, in the time that I've worked here, which is 7 years, we have had all of like 5 Shigellas. So most of the time, this is going to be a problem more if you go to places where sanitation and food services may be like reduced.
Mingle a little bit?
Yes. But yeah, very nasty. Okay, so Shigella, there are 4 serotypes that I want you guys to remember here. It may or may not be like a question on your test. I don't know. I don't know anything about that. This is my first time teaching this, so. But anyways, 4 antigens. So A is dysenteriae, B is flexneri, C is boydii, and D is sonii. And I've got some cheesy things to help you remember it. A, that's your primary pathogen. That's the one we think of, dysentery. B, B flexin. I don't know. C, this gets even worse because I think this came from your class. See you later, boy. Pretty sure Aaron came up with that. And then sonii, Sunny D is the way I think of it. So if it helps you remember those and kind of associate what serotype goes with what, please steal it. But if you've got better ones, I am completely open to it because I feel weird saying this every time. Cool. All right. And serotyping, this used to be a thing for you guys. It's not anymore where you have to know how to serotype a Shigella or a Salmonella. Now we just got to talk about it. But do you guys know what I mean when I say serotyping? Does that like make any sense to you? Or would you like me to explain?
It's like the latex part for the strep test, right?
Exactly, yeah. So we're just taking like a known anti-sera, a known antibody, and trying to attach it to an unknown bacteria to ID it. Is essentially what we're doing. And this was the procedure for it, but I don't care that you know it anymore. That is new. That's like last year that they decided we don't need this anymore. Okay, so Shigella, just ways that you can remember to group it. So how we are going to ID Shigella here. So on McConkey, it's going to be a non-lactose fermenter. On HE, it's going to be a non-lactose fermenter that also doesn't reduce H2S, so it's going to give that green color. Some other tests that we can use to kind of rule it in is its motility result. So remember, this is one of our keep still organisms, so it's going to be non-modal. But the crazy thing about this is that Shigella and E. coli biochemically and proteomically look very similar. Okay, so and just so you guys know, we use the MALDI for like next to everything. This is one of those blind spots of the MALDI where it actually can't ID a Shigella species, so we have to rely on biochems to ID it for us. Okay, so these are going to be the things that basically a table that would help you rule in what's an E. coli versus what Shigella. And the big one on here is going to be that lysine reaction. It's going to be, and it's a decarboxylation reaction, and we would expect Shigella to be negative and E. coli to be positive. There's some others on here of things that are variable, but when something's variable, that means like half of the time it's not the reaction that we would expect for it. So this is going to be the one that's going to be probably best.
Which one? Say that again?
Lysine decarboxylation. Cool. Shigella. All right, questions about Shigella? Okay, Klebsiella. This one's one of my favorites. I think it's got like a really interesting smell to it. I don't know. That sounds creepy, but
What does it smell like?
I don't know how to describe it. It's just like got a smell. It's like a smelly smell. I don't know.
Is it like one of those smells that some people like, some people don't, like gasoline?
Kind of. I guess that would be a good way of describing it. But it's like, yeah, it doesn't really like smell like anything. It's just got like this unique smell to it. You guys will know what I'm saying when you're on the bench. Don't look at me like I'm crazy, okay?
It sounds like a club candle that we can find for you.
I probably wouldn't smell good, but,
Yeah.
but I don't know. It just kind of like reminds me of bubble gum. Like it doesn't smell like gum, but it looks like bubble gum. That's just all. So Klebsiella, these tend to be a little more mucoidy than what our E. coli is. So our E. coli tends to be flat, remember, and Klebsiella tends to have sort of more of a boogery sort of appearance. They tend to be non-hemolytic on blood agar, and they are lactose fermenters, so you can kind of see them right here, real up and close, that they're really, really mucousy. And sometimes with some of these, they develop such a thick capsule around them that it actually slides off the plate. doesn't really stay there. So cool. So there's really two species of Klebsiella that we're concerned with, Klebsiella oxytoca and Kleb pneumo. Both of those are going to be the ones that we're concerned with. There are others. These are opportunistic pathogens in most places. They contain a capsule, most of them, and it's the K and keep still, so it's non-modal as well. They possess a potent urease enzyme. So do we remember urease? What infections do we think they have? Yeah, UTIs. But it can be also part of pneumonias and wound infections. Okay, this is like basically can cause really destructive like lung necrosis for like COPD patients and things like that. So Klebsiella. So this is going to be the main way we can tell the difference between the two. The indole reaction is going to be the best way to look at it. So based off of morphology, we can tell that it looks like a Klebsiella species based off of that mucoid appearance. And then we can do an indole to kind of speciate a Klebsiella pneumonia versus an oxytoca. Cool. Enterobacter species. All right. So on blood agar, it looks more or less like E. coli. It's not really a super mucoidy bug. Can be, but not always. But it's a large, flat, gray colony on McConkey agar. It is a lactose fermenter, but typically what we describe as a weaker lactose fermenter. It doesn't usually look lactose fermenty on the first day, but the second day, usually it looks like it is. Okay. Or the color can be really light like it is here on this picture. And so there's really two bugs here. So there's Enterobacter aerogenes and Enterobacter coachiae. I will tell you that this Enterobacter aerogenes has changed its name to now it's a Klebsiella aerogenes. Don't hate me. But this was back in 2018, so you might need to know that for boards. So it's technically a Klebsiella species now.
Which one?
The Enterobacter aerogenes. But for the longest time, they were still calling it Enterobacter aerogenes on boards. Okay. And this is one of those that's also an opportunistic infection. It's usually associated with contaminated medical devices such as respirators. And some of these can have capsules. And actually, I would want to change this, but most strains have inducible beta-lactamase production. So what that means is E. coli doesn't have any sort of known intrinsic resistance, meaning it's not going to be resistant to anything. But with these, it has what they call an AMP-C gene, which is something that will be resistant to most beta-lactam antibiotics just to begin with. So it's modal, but this would be the difference between an Enterobacter aerogenes and coachiae. The lysine reaction is going to be a good one. And the thing that makes it Marcia's favorite bug, that Enterobacter coachiae, is it's both ornithine and arginine decarboxylase positive. And she thought it was so convenient that the O and the A work right next to each other, and they were both positive here. So that's why it was her favorite. She's a very simple person, okay? Isn't that right, Robert?
Absolutely.
Absolutely.
Delay.
But that was her, that was why it was her favorite. Coachiae. All right. Pantowea. We'll do this and then we'll take a break. Cool. Okay. So Pantowea species. So I actually have one of these growing in a patient's blood culture that I'll share with you guys here in a little bit. We start talking about some IDs over there. But this is what it looks like. On blood agar, it's kind of got like this goldish sort of appearance to it. On our McConkey agar, it's kind of got like this, it's a non-lactose fermenter, but it kind of appears like a pinkish color. It's picking up the dye from the agar itself. But the big one that we're going to deal with is Pantowea glomerans. It was formerly an Enterobacter glomerans, but I would just know Pantowea as the name. It's pretty much non-reactive to everything, kind of hence the name Pantowea. It's not really reactive with much. And it's usually occurs from a trauma injury by things that have been contaminated by soil or water. So that's pretty much all I would know for Pantawea. It's one of those that doesn't really get talked about too often, but we get it occasionally. And like I said, I found it in a blood culture today. So I'm talking about that. Let me see it. Let's take like 10 minutes. Bathroom, run around, scream. I don't care.